because I want to make sure that we can get through a couple of these components. And I can always, if people want me to walk through a few of the other pieces, I'm happy to. But this is more of the high yield stuff. So when we think about antibodies, they're quaternary proteins. They have 2 identical heavy chains, 2 identical light chains. This allows for simultaneous binding to multiple antigens. And when we bind to multiple things, that increases avidity. Avidity means you're binding to more than one thing at once, versus affinity is how strong are you binding to that one thing. NPLEX tests affinity almost exclusively. They typically don't test on avidity, A-V-I-D, okay? The Y-shaped structure dictates the antigen binding, the effector function, and the flexibility. And certain structural questions often serve as a setup for functional reasoning here. Antigen specificity will come from interactions between our variable regions of heavy and light chains. So effector function, however, is mediated by the constant region, the Fc region of the heavy chain. So this is important to recognize. So our antigen specificity is the variable zone of the heavy and light chains, essentially up here, variable zone up here, versus the effector function, like what cell it's affecting, is happening from this Fc region of the heavy chain, okay? So the separation of function will help explain how antibodies can neutralize a pathogen, fix complement on the Fc region, promote then opsonization and phagocytosis. And so if we... Oh, sorry? Can you hear me? So changes in the heavy chain constant region is how we isotope switch, right? We go from IgG to IgM to IgA, et cetera, et cetera. That's an important piece to alter effector function, but we don't change the specificity of the antigen. So this is why you can start with an IgM antibody for some specific antigen, and then you can isotope or class switch to an IgG for that same antigen, because you're affecting just the Fc region, you're not affecting the antigenic region. Now, each antibody contains 3 hypervariable regions that are in both the VH, the heavy chain VL, the light chain domains. These are known as CDRs. So CDR 1, 2, and 3, which all together form your antigen binding site. The framework regions, the regions between these CDRs, maintain the integrity of the structure. So they ensure that the folding's done correctly. They ensure that the shape is the appropriate shape depending on the antibodies. They all have different shapes. On boards, mutations that affect CDRs are going to alter the specificity, what it's binding to, what the antibody is binding to. A mutation in the framework, the Fc region, will affect the binding entirely because it changes the protein shape. So if there's an Fc region mutation for an antibody, it's most likely not able to bind or work effectively. If there's a mutation that affects the CDR region, the antigenic region, it's most likely just not going to bind that antigen anymore, but it still might be able to bind something. It could potentially bind some of our tissue, right? It could be potentially an autoimmune condition, but that's an important distinction. Now there's 5 separate antibody isotypes that we recognize. So our IgAs, Ds, Es, Gs, M. This is all defined by the heavy chain constant region. So this like lower region down below here, being IgM being the largest. Let's see, what else is important to note here. NPLEX really loves to test on isotype-specific function. So things like IgA being mucosal immunity, IgE allergy, IgG opsonization and placental transfer, IgM the first one on the street. So if you have those function associations with your antibodies, most of the time you'll get through many of the NPLEX isotype questions for an antibody component. Okay. Now our antibody bound to B cell complex. The B cell receptor, or BCR, is not just a membrane bound antibody. It forms a signaling complex. So it's important to note, like this here, the B cell receptor, the BCR, right now is a B cell receptor, but it is an antibody. And if this B cell becomes a plasma cell, that means that we've released antibodies into circulation, right? So it's the distinction between B cell and our plasma cells. B cells have these B cell receptors, which look like antibodies because they are on their surface. Plasma cells have secreted their antibodies into circulation, but they come from B cells. Okay, so anyway, so B cell receptor forms a signaling complex. It forms that with Ig alpha and Ig beta. These are the, I'll circle them down here. So B cell receptor here, complexes with our Ig alpha and betas, these are stuck in the actual B cell membrane, and these contain ITAMs. ITAMs are just immunoreceptor tyrosine-based activation motifs. Yikes. Who wants to remember that? Essentially, it means that they're dealing with tyrosine, the amino acid, and that's what's inside here, these ITAMs, and that's the reactive component. That tyrosine can react with other things intracellularly and cause a secondary reaction, right? Cause an amplified reaction response. These motifs, these ITAMs, can transduce signals into the cell upon antigen binding, essentially cause a second messenger reaction inside of a cell. Accessory molecules are CD19s, are CD21s, are CD81s, CD anything, but CD2119 come up the most. These are signal amplifiers. They're going to either raise up the signal or sometimes they can block or blunt a signal. So you could think of them as like influencing what's happening with whatever's binding. This explains why BCR engagement alone can activate B cells under the right conditions. T cells, though, have to be co-stimulated to be activated. B cells can be activated on their own. They can be influenced to do other things. They could be upregulated or downregulated, but they themselves, if they bind to the right stuff, they can become activated and start to create an antibody. Now, our T cell receptor looks structurally similar to an immunoglobulin, but it's never going to be secreted. So that's a big difference between a B cell receptor. T cell receptor is permanently on the T cell. It's composed of an alpha and a beta chain as well. It has a variable and a constant region. It requires a CD3 complex to help it signal. So it requires something else to actually be able to signal, to be able to interact. And unlike an antibody, TCRs only recognized a processed and presented peptide antigen. So again, B cells can roam around, and if they find something they're sensitive to, they can bind to it on their own. T cells are divas, no offense. They have to have a packaged presented antigen via an MCH molecule, a major histocompatibility complex. So they cannot interact with a free antigen on their own. This is why T cell activation is much more tightly regulated. It has to be controlled. All right, so that T cell receptor, like I said before, structurally similar to an immunoglobulin. Functionally, it's distinctly different because it's restricted to any antigen that's presented on MHC complexes. The alpha and beta chain have some variable and constant regions. I think that's all I care about here. Oh, on NPLEX, if a stem emphasizes a mucosal surface, an epithelial barrier, or an innate-like T cell, the gamma delta version of a T cell, TCR, specifically T cell receptor, this here, is likely what they're referring to. Alpha beta is like our typical T cell receptor, the one that we associate with our adaptive immunity, but our gamma delta T cell receptors are more associated with innate immunity. You could think like they're closer to a natural killer cell than they are to an actual normal T cell. So if you saw anything like that, there's been some really strangely specific immunologic questions in the past couple of years. That's where this is coming from. When we look then at T cell receptor specificity as a whole, our classic kind of look is looking at our CD4 and our CD8 T cells. So when we're thinking about T cell receptors, they recognize antigens in context because it binds not just the antigen, but the antigen plus the MHC that it's bound to or presented to. So our endogenous or inside of a cell peptides, intracellular peptides, are going to be presented via MHC1 complexes, major histocompatibility type 1, and that's going to be on the surface or going to interact with our CD8 T cells. So our CD8 T cells, natural, or our T killer cells, our cytotoxic T cells, are only going to hang out or only going to associate themselves with MHC class 1. Therefore, those antigens that they're essentially being associated to or exposed to are going to be intracellular or endogenous antigens. Because what does a CD8 killer T cell do? It punches a hole through a cell that's infected, right? It's going to bore a hole through it. Now, our CD4 T cells is going to be communicating exclusively with our class 2 MHC molecules. That's going to be expressing our exogenous or extracellular peptides. And so outside of the cell things are going to be exposed to our CD4 T cells because our CD4 T cells are signaling T cells. Other things that could be important here, CD28 can provide co-stimulation or essentially upregulates or activates, while CTLA4 is going to block or break. So we could say what could increase an immune reaction, CD28, what can decrease an immune reaction, CTLA4. These are other co-stimulatory molecules that can be present or can interact with a T cell and can either upregulate or downregulate the response. If a T cell fails to activate despite recognizing an antigen, it's likely because there's a missing piece. Either CD28 is not present or CTLA4 is present and active and blunting the response. Okay. All right. What else do we want to talk about? We're going to talk about PAMPs. I scooched forward just one slide. So PAMPs are pathogen-associated molecular patterns. These are molecular motifs that are shared across multiple different classes of microbes. These could be lipoproteins from bacteria, flagellin, viral nucleic acids, fungal cell wall components. The immune system leverages PAMPs because they are hard for pathogens to mutate without actually losing fitness or competitive virulence. So PAMP recognition is not a part of our adaptive immunity. It's part of our innate immunity. So our innate immune system can identify PAMPs. It's going to act upstream then before our T's and our B's. It can help trigger cytokines, can trigger inflammation, and it can activate our antigen-presenting cells, which then can go present antigens to our adaptive immune system later on. On NPLEX, if you hear recognition of conserved motifs or rapid non-specific response, think PAMPs. PAMPs binding to a PRR, a pattern recognition receptor, that binding. Okay. These are some PAMP families. So toll-like receptors are one that's probably the most infamous. It shows where they're located, lots of different information about ligands and cellular functions. When you think toll-like receptor, think a membrane or an endosome, and it's going to recognize broad different microbial features like lipoproteins, nucleic acids, and its main thing is if it recognizes something, it's going to drive cytokine production and inflammation, also sometimes antivirals. CLRs are recognizing carbohydrates. So I think the C with carbohydrates, it's on the surface of stuff, oftentimes recognizes fungal components, and it's going to trigger phagocytosis and inflammation as well. RLRs and NLRs kind of hang out together because they're both found in the cytosol. And RLRs detect viral RNA, so the R and the R together, and that's going to activate interferons. Okay, so it's thinking some type of virus. Okay, it's fine. All right, the world's telling us to stop. NLRs are detecting bacterial peptidoglycan cell walls. That's going to cause a big anti-inflammatory response or a big inflammatory response. And then lastly, ALRs are detecting microbial DNA. So you'll see interferons and cytokines being produced. Classically, how I would summarize this, if a pathogen's viral and inside of a cell, think RLR or ALR as being who's going to respond to it first. What receptor is going to bind to something of that pathogen first. If it's fungal, think CLR. If it's bacterial, think toll-like receptor. So that's typically how I'd break those down. All right, I wanted to spend a little time on some of our cytokines. Cytokines and chemokines don't need to be confusing. They're just how the cells communicate, right? So we think of the endocrine system as communicating via hormones. The immune system communicates via cytokines and chemokines. The key component here is to try to pull out some like really big, broad, like this specific chemokine or cytokine does this specific thing from this specific cell. If you can connect one per cytokine, that will do you well. You don't have to remember every single thing about every single cytokine because that's impossible, but try to do like a 1-to-1 ratio, kind of like what we learned with bacteria earlier. So with our cytokine signaling, it's going to start with binding to a receptor. Typically, it's going to be high affinity and non-covalent bonding. Most cytokine receptors are going to trigger some type of downstream signaling effect that results in changes in transcription. Meaning that cytokines are essentially commands that are reprogramming cells, right, from that DNA level. Cytokine signal is any event that changes a cell's metabolic state, proliferation, differentiation, or behavior. So cytokines don't just turn on cells, they have really broad shaping effects. Three ways that cytokines can act, kind of similarly to hormones or other components, right? Autocrine, paracrine, or endocrine. So autocrine means that the cell that secreted it acts on that same cell. We could think T cells that secrete IL-2. IL-2 then is going to act on those T cells and encourage those T cells to proliferate, to expand, to grow, to divide. Paracrine is the thing that's secreted, it's acting on nearby cells. This is the majority of immune signaling. When in doubt, choose paracrine because it's almost always that in the immune system. And then endocrine, this is more hormones, right? But our endocrine enters the blood and acts systemically. Think of this as your big pro-inflammatory cytokines, your IL-1, your tumor necrosis factor, your IL-6, things that cause fever, systemic symptoms. Those are going to be endocrine. They're going to travel throughout the blood. Okay. All right. Three other kind of words that you should at least have some sort of brain recognition on is pleiotropy, redundancy, and synergy, just so you don't get tripped if you see these words occur. I haven't seen them tested on directly, but I have seen them used in a vignette to try to, or a question to try to throw students. Pleiotropy means one cytokine causes different effects depending on the cell. So IL-4 can promote a B cell class switch, but it can also help T cells differentiate. So IL-4 has two different actions depending on the cell it's interacting with. But it's the same exact cytokine. Redundancy means multiple cytokines that are different can produce the same response. So this allows for a compensation pathway. If like one cytokine fails to be produced, there's still another that can provide that said response. Synergy is two cytokines together have a greater than additive effect. So that's kind of the mechanism behind cytokine storm. Like multiple cytokines acting at the same time really add up and can create this amplification response. And then antagonism and cascade. Antagonism is essentially one cytokine will suppress or block the other's effects, kind of like pharmaceuticals. This is essential though for immune regulation, prevents inflammation from running away. So our anti-inflammatory cytokines, decreasing our pro-inflammatory cytokines. And then that cascade, this is one cytokine triggering a target cell to produce an additional cytokines that then amplify and diversify the response. This is oftentimes how a localized infection can escalate into a systemic infection. Now, cytokines are grouped in families, and I don't really care that you memorize the families. I just want you to know a couple key cytokines that are really important and come up a lot in NPLEX testing. So this is showing the families, and again, the only time that this can be helpful is because structurally sometimes they're doing similar things, but you're never tested on families of cytokines. So we're going to not focus too much on that, but I want to talk about a few cytokines specifically. Our IL-1 cytokines is this family. They're pro-inflammatory mediators. They are produced by macrophages and dendritic cells, and they're in response to a microbial antigen. Locally, if we think about what does IL-1 do, it's going to increase vascular permeability. It's going to encourage leukocytes to come to the site of infection. And systemically, it's going to tell our liver to produce acute phase proteins, and it's going to encourage fever. So that's your IL-1. It's your major pro-inflammatory cytokine. It does all the things. It's bridging our innate to adaptive immunity. It's promoting APC activation. It's helping prime our T cells. If you see fever and acute phase reactants, IL-1, IL-6, and TNF are your three culprits. IL-1, IL-6, tumor necrosis factor alpha, TNF. Lots of other stuff about cytokine IL-1. One thing that we sometimes see from a paired perspective with this class 1 of cytokines is defects in the common gamma chain, which is this here, associated with all of these cytokines. They all have this common gamma chain, YC or gamma C. This is a key for T cell and natural killer cell development and function. It's associated with all these cytokines, and there's a defect that can happen in this Y chain that causes X-linked SCID or X-linked severe combined immunodeficiency, and that's a pathology on your NPLEX exam. It's one that we don't think about because we don't see clinically. So if you see a defect or a mutation in the Y component of a cytokine, or really if you see a defect and a mutation in these cytokines, they're saying this patient has defect in multiple cytokines or multiple cytokines aren't binding appropriately because their FC region, their common region's not working how it should. think SCID, which is X-linked severe combined immunodeficiency. What's the classic kind of picture? You'll see T cell and natural killer cell failure. So failure of T cells and natural killer cells to do their job. From there, if T cells fail, your B cells will have kind of secondary B cell dysfunction because B cells are signaled partially by T cells. T cells help B cells class switch, right? So there's ultimate kind of dysfunction in our adaptive immune system. I think that would be the biggest piece there. YC chain mutation, impaired cytokine signaling, low T cells, low natural killer cells, B cells don't work. Okay. That's the only reason I brought up that slide. Your class 2 cytokines is all your interferons. Your type 1 interferons are your interferon alpha and beta. They're produced early by activated macrophages and dendritic cells in response to viral infection, or intracellular stuff. Their goal is to produce an antiviral state in neighboring cells by upregulating genes that promote ribonuclease activity, essentially promote things that break down viral RNA, inhibit protein synthesis to limit viral replication, and increase antigen presentation machinery. So essentially increase immune surveillance, like what you got hiding here, asking these cells, what do they have to show? So type 1 interferons protect our uninfected cells. They don't do anything to help our infected cells at all. They've just noticed, oh, this cell's infected. I want to make sure all my uninfected cells stay safe. So that's who they're acting on. They make them resistant to viral takeover. They're not just signals, they're reprogramming the cellular machinery. It's important to note that type 1 interferon alpha and beta, is different than type 2 interferon, or interferon gamma. This is distinctly different. So put them in two separate categories. Interferon gamma is activated by Th1, your Th1 type T cells, CD4 T cells. It also can be activated and produced by cytotoxic T cells and natural killer cells. So produced by 3 cell types, so it must be pretty important. It's the master cytokine of cell-mediated immunity, meaning it's going to be key component in activating macrophages, encouraging phagocytosis, and increasing reactive oxygen and nitrogen species. So if we think about the process of phagocytosis, interferon gamma is the primary kind of key driver or amplifier of that process. And it's thanks to our Th1, CD4 cells, our CD8 killer T cells, and our natural killer cells that it's produced. So if you see interferon gamma, it also is a good link for how adaptive immunity can still influence innate immunity. Because this is an adaptive immunity released thing, right? T cells and B cells, adaptive immunity, but it's acting on our macrophages, which is part of our innate immune system. So it encourages that cross communication. Okay. If a stem talks about an intracellular pathogen or any granuloma formation, think like tuberculosis, think interferon gamma as a potential cytokine that's involved. It's almost central to all those pathologies. Okay. That was that. Okay. Tumor necrosis. So tumor necrosis factor family is going to regulate development, effector function, homeostasis across not just the immune system. It's going to affect the skeletal and the nervous system. Tumor necrosis factor cytokines can be soluble or they can be bound to a membrane. They form trimers. They can bind together in threes, which helps them produce really effective binding to their own receptors. That's why it's really powerful, because it doesn't only have affinity, but it also has avidity, that multiple binding location, multiple binding spots. TNF signaling can produce or encourage cell survival, inflammation, or apoptosis. It's going to depend on the receptor it binds to, the context, and whoever is downstream that's kind of manipulating the response. So that's why TNF is hard, because TNF can do so many different things, tumor necrosis factor. Oftentimes, TNF questions hinge on systemic inflammation and endothelial activities like fever, hypotension, or shock. When we're comparing and contrasting kind of the two main players, TNF alpha and TNF beta are probably the ones that come up the most. TNF alpha is produced by activated macrophages versus TNF beta is produced by activated lymphocytes. TNF alpha is key pro-inflammatory cytokine, versus TNF beta is more involved in like cell-to-cell signaling, communication between leukocytes and endothelial cells. leukocytes, and the blood. So I think of TNF alpha as like pro-inflammation, IL-1, IL-6, TNF alpha, right? Those are your three pro-inflammatories. Versus TNF beta, it's produced by our adaptive immune cells, our lymphocytes, and is helping kind of then communicate to each other and communicate to the blood, communicate to our endothelial cells. Do we need more people? Do we need less people? Do we need the blood vessels to be more permeable or less permeable? Do we want to bring more fluid here or less fluid here? That's what TNF beta is helping to do. Okay. There's lots of others, but those are the two that you should know. IL-17 is probably the last one I'll talk about. IL-17 family consists of pro-inflammatory cytokines, and they're really at the kind of interface between innate and adaptive immunity. IL-17 receptors are primarily found on non-lymphoid cells, which makes them unique. They're acting on non-lymphoid, non-kind of immune cells, or non-lymphoid cells, not our T's and our B's. They're acting more on our innate immune cells, our neutrophils, or even keratinocytes or epithelial cells, cells that aren't even involved in the immune system directly. IL-17 is going to encourage neutrophil recruitment and activation, so cause neutrophil swarm. It helps with then defending against extracellular bacteria and fungi. We oftentimes see IL-17 signaling associated with chronic inflammation as well as autoimmunity. So we think about separating this out. It's pro-inflammatory. It's like IL-1, IL-6, and TNF alpha. They're all pro-inflammatory. IL-17, though, is more associated with chronic inflammation, so long-standing inflammation, keeping the process going, versus IL-1, IL-6, and TNF alpha are more acute. It's going to come on strong, and then it's going to resolve. IL-17 may then continue the party long-term if it's a chronic inflammatory state. That's how I differentiate those two. Okay. All right, let's see here. One pathology just to keep in mind, psoriasis is kind of a textbook example of an IL-17 driven chronic inflammation pathway. We have Th17 cells, so T cells that are Th17, T helper 17. They secrete IL-17A that encourages neutrophils to come to the site of activation, and that also activates keratinocytes. And so with both of those pieces happening, neutrophil recruitment and epithelial activation, we'll see that dysregulation in signaling leads to excessive keratinocyte proliferation because they're excessively being activated, and that leads to chronic inflammation at that skin surface level. And that's why you get these thick plaques, these rashes that are these thick plaques, because you essentially have big cell turnover, but you're not having adequate desquamation, not adequate release. You're having inflammation and buildup without adequate release of those skin cells. That's why oftentimes for psoriasis, we actually use IL-17 inhibitors. Oftentimes, if you'll see an IL-17 question on NPLEX, it's going to be extracellular pathogens, neutrophils, or autoimmunity. That's where you see IL-17 come up.